Hi everyone, I am Chidinma Onubogu, and today I'm going to be talking about a case study that is focused on a company called Aquapharm that is into the research of novel antibiotic treatments from the marine environment. So this is my presentation outline showing the various topics that I'm going to be talking about in this presentation. So first of all, what are antibiotics? They are substances that are produced by microorganisms that inhibit the growth of and even kill other microorganisms. By this definition, it means that antibiotics do not include other antimicrobial substances that are synthetic or semi-synthetic or come from plants and animals. Therefore, we can say that all antibiotics are antimicrobial substances, but not all antimicrobials are antibiotics. Now I'm going to be talking about the discovery of antibiotics. The first antibiotics that was discovered is penicillin. It was discovered by Sir Alexander Fleming in 1929, and it was introduced into clinical practice for the treatment of Staphylococcus aureus in the 1940s. However, in the mid-1940s, just few years after its introduction, Staphylococcus aureus showed resistance to penicillin. This was the beginning of the era of antibiotic resistance. So, what does antibiotic resistance mean? It is simply the ability of bacteria to resist the effects of an antibiotic they were once susceptible to. It means that bacteria that could initially be killed by a particular type of antibiotic can no longer be killed by the same antibiotic. The Center for Disease Control and Prevention declared in 2013 that the human race is now in the post-antibiotic era. The World Health Organization warned in 2014 that the antibiotic resistance crisis is becoming there. Now I'm going to be talking about the mechanisms by which antibiotic resistance occurs. The first is by inactivation of the antibiotic. This can be seen in the diagram where the bacteria produces drug inactivating enzymes to cause inactivation of the antibiotic. The next is by reduced permeability of the membrane to the antibiotic. This can also be seen here. The cell wall becomes modified, inhibiting uptake of the drug. The next is by increase in the efflux of antibiotic from the cell. This can also be seen here. The efflux pumps are activated. Efflux simply means flowing out of a substance. So the efflux pumps are activated and the antibiotic is caused to flow out of the cell. The next is by altering the site of action of the antibiotic. This can be seen here by alteration of drug targets, and all these are the mechanisms by which antibiotic resistance occurs. Now I'm going to be explaining the causes of antibiotic resistance, and the first is by overprescribing of antibiotics. When we have an infection that does not necessarily need antibiotics, the doctor prescribes antibiotic, and this contributes to antibiotic resistance. The next is by patients not finishing their treatment. When patients do not finish their treatment, the resistant bacteria in our system multiplies. The next is by overuse of antibiotics in livestock and fish farming. Most farmers use antibiotics to obtain better quality and healthier animals. The next is by poor infection control in hospitals and clinics. Studies have shown that most antibacterial resistant infections occur and spread in hospital settings. The next is by lack of hygiene and poor sanitation. And finally, the lack of new antibiotics being developed. Now I'm going to be talking about antibiotic-resistant infections in the world today. The Center for Disease Control and Prevention carried out an assessment of antibiotic-resistant threats, and based on their results, they divided or classified these threats into three: the urgent threats, the serious threats, and concerning threats. Urgent threats are high-consequence antibiotic-resistant threats. They may not currently be widespread, but they have the capacity to become so, and they require urgent public health attention to identify infections and to limit transmission. The serious threats are significant antibiotic-resistant threats. They are not considered urgent, probably because of low domestic incidence, or because there's reasonable availability of therapeutic agents for them, but they have the capacity to become urgent without ongoing public health monitoring and prevention activities. The last one is the concerning threats. This basically refers to bacteria with low antibiotic resistance or with multiple therapeutic options. These threats require less monitoring and prevention activities. Based on this assessment, it is obvious that the world is in serious need of new antibiotics to deal with these resistant infections. This has brought about the need to explore other sources in our search for new antibiotics. The video in the next slide explains why we need to look to the oceans in our search for new antibiotics.
For the past 5 years, we've been looking very carefully at a variety of marine sources for their production of natural antibiotics. The vast majority of all of the drugs to treat infectious diseases are from nature. Over 120 antibiotics were extracted from soil bacteria, a very productive activity. So we obviously are adapted to study terrestrial resources and have done so for many, many years. But now consider the ocean. The microorganisms in the ocean are different genetically. They tend to produce different kinds of molecules. We bring these organisms back to the laboratory. We isolate pure strains of bacteria, and then we grow them in smaller scale and test to see if any of those microorganisms can produce antibiotics. And we found a significant number, at least 5 in the last 5 years, that I think are of interest, potentially could be developed into life-saving drugs. And one of those discoveries is a chemical compound we've called anthracimycin. It's a very potent antibiotic. It is a unique chemical structure type and has activity against some of the most dangerous drug-resistant pathogens like Staph aureus, so-called MRSA, which is the methicillin-resistant staph, perhaps the most difficult infectious agent to cure. The number of drugs that are, in an overall sense, that are coming from the ocean is increasing dramatically. There's 7 drugs on the market from the ocean. There's 26 drugs in clinical trials, including some from my lab. So the prospect of finding antibiotics is extremely high, and we need to begin to recognize what this source, the world's oceans, represents.
So these are the steps involved in the marine drug development process. It begins with collection and identification of marine organisms from the oceans, and then cultivation if necessary. If we need to get enough samples for further analysis, then we need to carry out cultivation. The next is the extraction of the bioactive compounds, screening, which could be biological, chemical, genetic, and then identification and selecting of interesting extracts. Basically, the rest of the steps involved in the process, all the way to registration according to valid legislation, are the same steps that are involved in drug development from terrestrial organisms. However, it is interesting to note that there are already marine drugs on the market. So I'm going to be talking about the marine drugs on the market and their therapeutic area. The first is Adcetris. Its natural product was isolated from a sea hare, and it is used in the treatment of Hodgkin's and large cell lymphoma. Cytostar U is also called cytarabine. Its natural product was isolated from a marine sponge, and it is an anti-cancer chemotherapy drug that is used in the treatment of leukemia. Hyaluron, its natural product was isolated from a Japanese sea sponge, and it is used in the treatment of metastatic breast cancer. Yondelis, its natural product, was gotten from a Caribbean sea tunicate, and it is used in the treatment of advanced soft tissue sarcoma. So the first four are used in the treatment of cancer. The next one is Caragellos. It was isolated from red seaweeds, and it is an antiviral drug in the form of a nasal spray. The next one is Vira A. It was isolated from a marine sponge. It's an antiviral drug that is used to treat herpes viruses, pox viruses, and certain other types of viruses. Lovasa was gotten from fish oils and is used for the treatment of patients with hypertriglyceridemia. This means high levels of triglyceride in the bloodstream. Triglyceride is a kind of lipid. So Lovasa is used for the treatment of this condition. The next one is Prialt. The natural product was isolated from a cone snail called Conus magnus, and Prialt is an intrathecal drug, which means it is administered into the spinal cord, and it is used for the treatment of neuropathic pain. It was the second neuropathic pain drug to be developed after morphine. The case study is focused on a company called Aquapharm Biodiscovery, whose core focus is to develop antibiotics to treat gram-positive and gram-negative infections such as Pseudomonas aeruginosa, Acinetobacter, methicillin-resistant Staphylococcus aureus, penicillin-resistant Streptococcus pneumonia, and vancomycin-resistant Enterococcus. Their other focus is to isolate novel marine products for pharmaceutical applications. It is worthy to note that Aquapharm already has lead candidates, with one of them in the preclinical development stage. AQP182, which is the one in the preclinical development stage, is believed to be effective against a broad spectrum of multi-drug resistant bacteria, such as the multi-drug resistant Staphylococcus aureus. The other candidate, P1639, has shown promising results in suppressing NFKB. Now, NFKB is a transcription factor that is found in mammalian cells, and it is involved in our cellular response to stimuli, and it plays a major role in regulating the immune response to infections. P1639 is being assessed for personal and consumer healthcare applications. Nevertheless, there are challenges faced in the discovery and development of marine natural products. And these challenges have been grouped into three: biodiversity challenges, supply and technical challenges, and market challenges. Under biodiversity challenges, biodiversity simply means biological diversity, which means variability among living organisms of all sources. So the first challenge is the sampling techniques. Now, because there is no traditional knowledge about marine natural products or marine organisms, sampling has always been done by random selection. And in obtaining these samples from the marine environment, scuba diving is usually done, but scuba diving cannot be used to obtain marine organisms that are deeper than 30 meters in the oceans. And so trawling is used. However, trawling is inefficient because it destroys the samples and it also destroys the ecosystem. The other challenge is the insufficient information on taxonomic identification and classification, inefficient microbial libraries. For supply and technical challenges, the first and major challenge is the permanent availability of adequate amounts of organisms and compounds without causing harm to the environment. This is the supply challenge. The next is the variability of the marine natural products producer. Now, due to variability in environmental conditions, it is possible that a resupply of the same organism will not produce the same metabolites. The next is proper identification of compounds, understanding the mode of action of a lead compound against a specific target. And under the market challenges, we have identification of market need, identification of price tag per unit, which is supposed to be supported by markets, identification of production technologies that supports the possible price level, and then we have identification of the volume needed for final manufacturing and supply, regulatory requirements, and the lack of adequate funding from the government or international organizations. However, there are solutions to the challenges faced. For biodiversity challenges, sampling strategies can be optimized by using sophisticated engineering technologies, improved taxonomic expertise, screening of pre-fractionated and crude libraries simultaneously, and carrying out integrated analysis of the results. For the supply and technical challenges, total chemical synthesis and microbial fermentation can be used to increase supply. Application of biotechnology and identification of the true producer of a secondary metabolite from a symbiotic assemblage. Metagenomics approach can be used, as well as sophisticated nuclear magnetic resonance spectroscopy to identify natural products. Application of the knowledge of molecular biology as well as assay development can help in determination of mode of action. For the market challenges, market guidelines need to be incorporated in marine discovery programs. Collaboration of academic researchers with industry partners. This is very important as it brings together the academic researchers' deep knowledge of marine life with industry's development needs, market awareness, and business expertise. The next solution is the production of cosmeceuticals as a means to generate funds for development of pharmaceutical application. Natural products discovered can be used for production of cosmeceuticals, as studies have shown that it has a faster route to the market. An example is a cosmetic product called Refermer, whose natural product is known to also have pharmaceutical application. However, while researchers are out there trying to find new sources of antibiotics to deal with the resistance crisis, we as individuals need to play our part in controlling antibiotic resistance by proper use of antibiotics only when they have been prescribed and when they are needed, finishing our prescribed antibiotics, and finally, we need to take public health practices seriously. We need to keep our environment clean, and we need to imbibe the culture of personal hygiene. In conclusion, it is known that the oceans occupy 70% of the Earth and is the largest ecosystem on Earth. It is characterized by unique growth conditions and encloses massive biological and chemical diversity, thereby providing a vast opportunity for discovery of novel therapeutics with novel mechanisms of action. There are currently 8 approved marine-derived drugs on the market, and there are various compounds in different phases of clinical development. Innovations in several fields as well as advancements in technology have been used to overcome some of the hurdles that are associated with marine drug discovery and development. Therefore, I am optimistic that the marine environment will provide a new wave of drugs that will stem the tide of antibiotic resistance. Thank you.